welcome to the keynote address of Research Day. I'm going to hand it over to Anushka in a moment, who's going to introduce our keynote speaker. In the meantime, I think we've had a great day so far. Nice posters, really good platform presentations. So as I said at the end of the platform presentations, I think our graduate students really make us all proud. So thank you all for doing a great job. All right, so Anushka, take it away.
Good afternoon, everyone. It is my pleasure to introduce our keynote speaker, Dr. Laufey Amundadottir. Dr. Amundadottir received her Ph.D. in cell biology from Georgetown University in 1995 and completed her postdoctoral training in the Department of Genetics at Harvard Medical School. She later joined Decode Genetics as the head of the Division of Cancer Genetics, where she led genome-wide linkage and association studies across multiple cancers. In 2007, she joined the National Cancer Institute and became a tenure-track investigator in the Laboratory of Translational Genomics in 2008. She was awarded scientific tenure by the National Institutes of Health and appointed senior investigator in 2017. Her work has been recognized with several NCI Intramural Research Awards, a Center of Excellence in Integrative Cancer Biology and Genomics Award, and the NCI Women Scientist Advisors Mentoring and Leadership Award in 2019. Please join me in welcoming Dr. Laufey Amundadottir.
Thank you so much for the really nice introduction. And thank you all for inviting me. It's really been a pleasure to spend the day with you and just learning about all the interesting science that you're doing here, whether it's cancer science that I know a little bit better, or neuroscience that I've learned a lot about today. So just really interesting work that is ongoing. So I wanted to tell you a little bit about the work that I have been doing at the NCI. And I think I may not need too much of this, but just to give some extra as to my background. So I'm born in Iceland, and I went to school there until I graduated with my undergrad from the University of Iceland. A very small school, but it was really nice to be there. Then I did my PhD at Georgetown, working on animal models and breast cancer. And after that, I moved to Harvard to continue my animal model work, again in breast cancer and focusing on signal transduction. Then I was recruited to Decode Genetics, which is a gene mapping company in Iceland, really using the fact that the population is really small, genealogy is really well known, and the records of cancer are very complete and go all the way back to 1950. So with genealogy and patient records being very strong, doing genetic mapping can be really powerful. And then finally, I was recruited to the NCI in 2007. It's amazing that it's been that long, almost 20 years. But at the NCI, I lead studies on genetic mapping of pancreatic cancer. So just to start with, what is gene mapping? I know from the posters that some of you do gene mapping, but in reality, it's really the correlation of genetic variants in our genomes to phenotypes. And those phenotypes can be diseases, it can be cancer, diabetes, whatever disease one is interested in. But it can also be more linear phenotypes, so height, body mass index, etc. Or it can be responses to say, drugs, etc. So in a way, pick your phenotype of interest, and then you can do gene mapping. And this we all know, the human genome, our 22 plus XY chromosomes. There are about 3 billion nucleotides in the human genome and about 78,000 genes. So you can count them differently, whether you count coding genes, non-coding, etc. But for gene mapping, we of course focus on the genome, and what we're looking at is inherited variation. And our genomes are I think 99.99 something identical, but the variation in our genomes, whether that variation is in the coding regions of the genes or in the non-coding parts of our genome, that is what allows us to map genes and map diseases. So I'm sure that there are actually more than 1 billion DNA variants. Some of them are really, really infrequent. Some of them are variants that we call private, so just seen in one person in the whole world. But what we use for gene mapping are more common variants. So in terms of what kind of variants there are single letter variations, what we call single nucleotide polymorphisms, or SNPs. And you'll hear me talk a lot about SNPs today. So really what they are, they're just one letter differences between people at one specific location in the genome. There can also be small insertions or deletions. Here I'm showing an insertion of A in that sequence. There can be repeats. So here I'm showing a TAG, so a trinucleotide repeat that is repeated 3 times in the top row, but 2 times in the bottom row. And then we can have inversions. So because I'll be talking mostly about SNPs, I wanted to introduce you to how we call them. We usually have numbers that are RS and then followed by a number. And this is just the naming convention. I think it stems from the early days when the same SNP could have more than one number, and then they were combined into these RS numbers. So I talked about inherited variants. So that's very important for gene mapping. I talked about phenotypes, but then recombination, basically the recombination that happens in early meiosis is really important because that gives us this correlation between variants that we're using. And you all know this from prophase in meiosis 1. And you can see here that the capital letter A and B variants, after recombination, they're not close to the C, but to the uppercase C, but to the lowercase c. So this correlation is basically seen all over the genome, and what directs sort of how closely or poorly genetic variants are linked is both how close they are on a chromosome, but also because of just population history. Your parents, their parents, influx into populations, etc. So these two concepts are really important. And recombination basically gives us linkage disequilibrium, which is defined as the non-random association of alleles at 2 or more loci. So the alleles for a SNP would be, say, A or T or A or G or what have you. And this is what is really important for gene mapping. Here I'm showing you 2 SNPs. And again, these are variants that have 2 different alleles or 2 possibilities. So here I'm looking at SNP1 that has a C or a G allele, and then the options are just 2, C or G. SNP2, an A or a T SNP. And we have A linking up with C, T linking up with C, etc. And when we talk about haplotypes, it's really the combined alleles at 2 or more loci. So as you can see this, and this is of course just a made-up example, the CA haplotype, you see that in 35%, and CT 22, GA 26, GT 17. So these SNPs are in frequency, or these haplotypes are in frequency close to 25% of each, and that tells us that there is low LD between these 2 variants. However, on the other side, we have 2 SNPs that are in high LD. Again, we have 2 SNPs, SNP1 and SNP2, but if you look at the frequencies, we have around 50% of the AC and almost 50% of the GC. So really, what you can tell yourself is that if you genotype, or if you have information about the alleles for SNP1, you almost know without doing anything what is happening at SNP2. And this is what we use both for gene mapping, but also for imputation, which I'll come to a little bit later. But this really helps in terms of gene mapping. And the way we often look at LD is that we mark SNPs or variants in high LD with a dark red color. And when there is low LD, we mark it with white or just a sort of a more faint color. And this, again, helps us in gene mapping. And I'll explain that a little bit more later. So going back to the disease that I study, the pancreas is one of the very important digestive organs. It has sort of two compartments. It's like a little strange organism. It's a two-in-one organism. The exocrine pancreas has acinar and duct cells, and you can see them, the acinar cells here on the histopathology slide, and the duct cells here surrounding a duct. But it also has the endocrine pancreas. So the exocrine pancreas is very important for digestion. It creates and secretes massive amounts of digestive enzymes, some of them listed here. But the endocrine pancreas has more to do with glucose metabolism and glucose homeostasis in the organ. So even though there's cross-talk between these two compartments, it's really sort of the function is quite separate. And what I study is what we call pancreatic ductal adenocarcinoma, which is the most common pancreatic cancer. The endocrine pancreas cancer arising in that part of the pancreas is only about 5 to 10% of all pancreatic cancers. So even if I say pancreatic cancer from now on, I'm really talking about pancreatic ductal adenocarcinoma, or PDAC, as we call it for short. So some background, pancreatic cancer is not one of the most common cancers there is, but it's really high up there in terms of mortality, and mortality has been going up. So now it's the third most common cause of cancer, but in a few years, it's predicted to become the second most leading cause of cancer deaths. And the reason may be because it's hard to get to it. There are no sort of easy symptoms. And usually when patients and doctors realize what's going on, it's too late. So it's really, in terms of mortality, it's one of the worst ones that is out there. In terms of risk factors, so age is a risk factor for all cancers, but beyond that, smoking, diabetes, obesity, heavy alcohol use, and then chronic pancreatitis are well-known risk factors. I don't know if people know what chronic pancreatitis is, but it's really an inflammation of the pancreas. It can be inherited, or it can be sporadic, so just come in episodes and then disappear. Family history for pancreatic cancer is also a strong risk factor, and sometimes cancer occurs very sort of dense in families, and we often talk about those families as sort of families with cancer syndromes, and often they're explained by mutations, or they're inherited, so not sporadic mutations, but mutations in coding regions of genes. Think about BRCA1, BRCA2, ATM, etc. And those mutations often increase the risk of some of these syndromes quite dramatically. So this is sort of the landscape of genetic variants that increase risk of pancreatic cancer. On the x-axis, you have variants going from rare to quite common. On the y-axis, we have the effect size, so how increased is the risk, going from low to high. So here's rare variants, and we say familial disease or Mendelian disease or cancer syndromes when we talk about that. On the other hand, common variants with low effect sizes tend to be identified by GWAS. The other circles, low frequency variants with intermediate effects, they can be identified through approaches that we usually use for familial cases, or sometimes through GWAS. Common variants with high risk are not very frequent at all. They're seldomly found. And then rare low-risk variants are out there, but they're not easy to find because you really need very high sample sizes. So bringing this back to pancreatic cancer, in the section here where we're talking about rare high-risk variants, I've listed some of those genes that I already mentioned, but beyond those, we have genes that are important for inherited forms of pancreatitis. So these patients that carry mutations in these genes have dysfunction in the production of digestive enzymes. And often what happens is that the enzymes are activated before they leave the pancreas to go into the gut. And that can cause really bad bouts of pancreatitis, and it increases risk of pancreatic cancer quite dramatically. So then, and again, this is familial, and usually when you're studying or trying to find these genes and mutations, you study families. What I focus on the other hand, is genome-wide association studies, where you're really looking at populations and a big number of cases and controls. And they're common and tend to be low risk, so very different from the familial mutated genes. Let's see, what am I doing? Okay, so GWAS study design. How do you go about doing a GWAS? First, you need a representative sample of the population, and good studies to use are cohort studies that are big studies where you start with a large number of people, you follow them prospectively, and whoever develops a disease that you're studying becomes a case, and then you select controls from the rest of that population. But this can also be case control studies, case series, family-based studies even. Then you need to genotype the whole genome, so the whole 22 plus the X chromosome. Usually these arrays don't have variants on the Y chromosome, or if they have, not very much. So initially, these genotyping arrays were smaller with about 200,000 or 300,000 SNPs. Now, the most frequently used arrays have about 1 million SNPs, so say 700,000 to 1 million SNPs. What we're testing, sort of, if you think back to the graph that I showed on the last slide, we're really looking at common variants. So usually their frequency is above 5%. If the study is bigger, we're looking at things or variants that have a minor allele frequency of more than 0.5%. But it all depends on the size of the study and therefore the statistical power. QC is really important for GWAS, and I'll come back to that a little bit later. And really, we're testing so many variants, meaning that we're testing a lot of hypotheses. So we cannot use a p-value of 0.05, like when we're testing just one hypothesis. So we use something called the Bonferroni correction of our testing, and usually our threshold is for significance is that the p-value has to be lower than 5 times 10 to the minus 8. And we tend to validate things by replicating again and again until we believe what we see. Also, we tend to use reference samples because population stratification is very important, and it can lead to findings that are spurious if we don't carefully match our controls to our cases. So QC and all of that takes months. It's not fun, but it's really important to make sure your data is good. So here's basically what we do. We use these arrays to genotype controls and cases. We actually usually after genotyping, actually always nowadays, we impute. So having 1 million variants on the genotyping arrays is not enough. And there are methods where you can take reference samples and whole genome sequence data and use that haplotype information that I talked about earlier to basically guess or infer genotypes at missing variants. So we tend to do that. And what we get out is basically here on the x-axis, we have chromosomes, and every dot is a SNP. So chromosomes from beginning to end, from 1 up to 22 and X. And then on the y-axis, we have the significance. And we usually do negative log p-value just because then our peaks are the most significant findings. So this is how it's done. Getting back to QC, so populations from different countries and different continents can have variants that differ in frequency. So we usually use reference samples to look at population. We call it population structure. And this is just, in a way, a similar method to a principal component analysis. But here you can see the European ancestry samples here. Really, we're just measuring similarity to reference populations. Similarity to the African reference sample populations here on this axis, and then to the Asian reference populations here. And if we go even more fine-grained, this is really a PCA plot of the genotypes. Here we can see the samples with European ancestry, African and Asian ancestry, and then over here, we can see even within Europe, slight differences that the PCA analysis can pull out. And if you think about it, if we had cases from Finland and the controls from, say, Denmark or France, we would get really strange results that are just from population stratification. So it's just really important to make sure we always match cases and controls really well. So then going back to the study that I co-lead with people within the Pancreatic Cancer Cohort Consortium in collaboration with the Pancreatic Cancer Case Control Consortium. So the PanScan study that I started working on almost 19 years ago is really a cohort of cohorts. So it's really a lot of cohort studies that came together to study not only pancreatic cancer, but multiple other cancers. And of course, pancreatic cancer is not very common, it's deadly. So to get the number of cases and controls that you need for a well-powered study is hard. So very quickly, we moved over to incorporating just any study that we could get our hands on, because numbers and statistical power is really important. We also collaborate with the Pancreatic Cancer Case Control Consortium, and their studies are listed here. And then we've collaborated with a number of studies in Europe, biobanks, companies, the company that I used to work for before I joined the NCI. And more recently, I've set up studies in Korea with 10 different hospitals in the Seoul area. We collaborate with groups in China, in Japan, who have already produced and published studies and are willing to provide data for us to put together. So what I'll talk mostly about today is a study where we had about 9,000 pancreatic cancer cases and about 13,000 controls. What we have ongoing now is a much bigger study that hasn't been published, and it has over 37,000 cases and about 1.9 million controls. So a much better powered study than what we had before. So this is from the smaller study, so the 9,000-ish cases. So from that study, in a number of different phases, we identified just over 20 pancreatic cancer signals. And what they basically are, at the beginning when you identify them, they're just regions on the genome where you have SNPs that are highly correlated, usually not one, unless, like if you had a case control set of 3 million cases and 5 million controls, you might have the power to reach just one variant. But usually we have a number of highly correlated variants. So at the beginning, these are just landmarks on our chromosomes. We've used a technique that's called a transcriptome-wide association study, where we have data that has gene expression and genotypes that we can infer and kind of impute into our GWAS data set. And using that approach, we basically find genes where their genetically regulated gene expression is different between cases and controls. And as you can see, some of these genes are at loci that we had seen before from the GWAS, but some are at completely new loci that we hadn't seen before. So these two approaches can be used for gene mapping. And again, just sort of bringing home the message, these are all quite common variants. So minor allele frequency of all of those are above 5%. In our newer study, we're finding signals that are less frequent. And that's not surprising because our power is much, much better. The odds ratios, it's really important to stress that what we find through GWAS is like if somebody has a risk allele at one of these loci, it's really the risk increase is only about, say, 10 to 50% above the population that does not have that variant. So the risk for one of these is really nothing to do anything about. But when you combine them, especially in our new study where we found a lot more, the combination of risk alleles can be put together into something called the polygenic risk score. And you can use that for prediction, like who is more likely to potentially be diagnosed with pancreatic cancer. But at this point, it's really not very useful. What I've done in the lab is to try to understand the molecular mechanisms of some of those. And I'll show you two examples of what we've done. In sort of contrast with the familial mutations that tend to be coding, what we tend to find is mostly non-coding. So we have to go and try to figure out, of these non-coding variants, are they in promoters or enhancers? Do they slightly affect, say, binding of a transcription factor, and then thereby influence expression of a gene that's either nearby or even at a distance? So there's a lot of molecular work that happens. This is actually from our new study that I call PanScan-4. So just over 37,000 cases and actually close to 1 million controls. About 30,000 of the cases are of European ancestry, and about 7,000 are of East Asian ancestry. Actually, there is a small component of African American cases and controls, and we're hoping in the future to increase that part even better, because we feel it's really important not only to focus on the European ancestry individuals, and things might be slightly different in individuals of different ancestries. But so in this new study, instead of just over 20 loci, we now see 119 loci. And just to tell you the truth, that going through these and trying to sort of understand these loci is really a headache, but it's a lot of fun at the same time. We're seeing genes close to some of these loci that are important for pancreatic development. We actually do see a signal not far away from KRAS, which is really important for pancreatic cancer when it's mutated somatically. So we feel that there's a lot of new exciting information in this data set. Okay, so target genes and molecular mechanisms. So through the work of people in my lab, we have identified functional variants among these highly correlated variants at each locus and target genes at these loci here. For some of them, we know more. For others, we know some parts. We think it's the gene, like ABO, for example. It's the gene that dictates what blood groups we're of. It looks like that may be the underlying gene at this locus, but we don't fully understand the biology. Some of these genes where we've identified the target gene, like this gene here encodes a protein that's part of the ubiquitin ligase kind of degradation complex. NR5A2 is important for pancreatic development, but also for homeostasis. So if the pancreas is inflamed, this gene is upregulated when it's brought back to homeostasis. PDX1 is really important for pancreatic development as well. It's also mutated in some forms of type 2 diabetes. This 3 is a protein that regulates RNA processing and RNA amounts. TERT is a gene that you may well know. So this gene encodes a protein that regulates telomerase. Actually, that is part of telomerase and regulates the ends of our chromosomes. It's very often mutated in cancer. CLPTM1L was a less well-known gene when we started working on it, but it's actually through our work and through the work of others, we've shown that it's upregulated in pancreatic cancer, in lung cancer, and some other cancers. CTRB2, that locus, 16q23, is a locus that I wanted to tell you a little bit more about. So CTRB1 and 2 encode for chymotrypsinogen precursors, proteins that are really important for for digestion. So when we saw that signal, we have again that RS number. So this is a SNP with just 2 alleles, AG, say, or something like that. The GWAS p-value was 7.6 times 10 to the minus 17. Odds ratio, if for individuals carrying either 1 or 2 copies, is 1.33. So the risk over the general or people without the risk allele is only 33%. So it's not hugely strong at all. But we were interested in knowing what was going on. And here I show you where that SNP that marked that signal is located. CTRB2 is downstream, CTRB1, and here another gene that's important for breast cancer. So when we have these signals, we start by what we call imputation and fine mapping, and then we statistically decide or define what is the set of variants that we're 95% sure that we'll find a functional variant. We can never be 100% sure, but this is usually what we do. So we noticed 9 other variants, and we decided these are our variants. Now we'll go in and look for the variant that is basically underlies the signal. And when variants in a signal are all non-coding, we tend to think, okay, this one of them is in an enhancer or a promoter. This is really gene regulation. So we start looking at epigenetic annotations to see if any of these like overlap enhancers or histone marks, et cetera. But then we also correlate genotypes to gene expression. So when we did this for this locus, we found nothing. So basically 0, which really surprised us. But then we decided to look further than just correlating the risk alleles to gene expression. We looked at splicing. And as you can see here, on the x-axis, we have samples with different genotypes. So C is the risk increasing allele, G is the non-risk increasing allele. And CTRB2 has 7 exons, and usually, as we all know, splicing goes from exon 1 to 2 to 3, 3, 4, etc. But we saw this really strange splicing effects where exon 5 to 6 and 6 to 7 splicing was way down in individuals with the risk increasing allele. And then we saw, is it me who's creating these noises? Oh, it's my hair. Okay, I don't have anything to put it here. Let's move it back. But we saw exon 5 to 7 splicing. So basically, this splicing was high in people with the risk increasing alleles and really telling us that somehow splicing is going over exon 6, so somehow skipping exon 6. And we saw this in the GTEx consortium data. I don't know if you know about it, but it has expression data from like multiple different tissues, and this is all normal tissue-based, and it has also genotype data. So you can do these studies, whether you're correlating your genotypes to gene expression or splicing or isoforms. This is really a good data set. We had our own smaller data set that we replicated this finding in. And then we did a so-called co-localization. So we statistically asked the question, is the GWAS signal and the splicing QTL signal the same signal? And we got a very strong indicator that splicing was indeed co-localizing with the GWAS signal. So we then looked at the literature, of course, asking ourselves, like, why is exon 5 splicing to 7 and just skipping 7? And we came across this paper that had looked at deletions and insertions in the genome, and they had described a deletion variant in different populations that overlaps exon 6. So we felt this might be an explanation for why we see exon 5 to 7 splicing. What would happen to the protein is that we would basically skip exon 6, and there is a stop codon early in exon 7. And of the 3 active sites in CTRB2 in the protein, this one would be completely gone. So we started out thinking this is non-coding, this is regulating gene expression, but it really led us to something that's coding that really kind of destroys the protein, most likely. So here we set out to first ask the question, is this deletion, can it be responsible for that signal? And is it the same, should it be a member of that set of 10 variants? So we used PacBio long-read sequencing. We sequenced a number of reference samples, and then we imputed that deletion back into the GWAS. And basically, we saw that the odds ratio was very similar for heterozygous individuals, somewhat higher for homozygous individuals. But then with additional work, we basically showed that if you adjust for the deletion, the rest of the signal goes away. And if you adjust for the top SNP of the signal, the deletion signal goes away. So really telling us this is the same thing. And then we were curious to know what happens to the CTRB2 protein when you have that deletion. So we first looked at activity, and we overexpressed both wild type and the exon 6 deleted CTRB2 in cells in vitro. And we saw that full length has very nice, strong chymotrypsin activity, but the deletion really, almost all of the activity was gone. And then, like I told you earlier, the enzymes that are produced in the exocrine part of the pancreas, they're secreted into the gut where they have their function. So we also looked at secretion. And if you look at the full-length CTRB2 here, there's not a lot in the cell lysate. Most of it is secreted into the media. However, for the exon 6 deletion, a lot of the CTRB2 protein is inside cells. So the pancreas basically produces massive amounts of these proteins, and having it sit inside cells is probably not good. Not only do you lose the function, but you may cause some damage to the pancreas. So here we showed by immunofluorescence that the truncated, the exon 6 deleted CTRB2 protein is retained in the ER, the endoplasmic reticulum. Here we have an ER marker. We have our CTRB2, either full length or deleted. And if you co-localize with an ER marker, you can see how much of it is retained in the ER when that deletion is there. So having something accumulate in the ER probably leads to ER stress. So we looked at publicly available data and compared samples with 0, 1, and 2 copies of the deletion. And we basically, the most significant pathways were those involving ER stress, the unfolded protein response, which is important in terms of helping the ER if proteins are not folding normally, then this mechanism kicks in, and translational pathways. So this probably affects translation and definitely causes ER stress. We also co-localized the deleted version of CTRB2 with ER stress markers. Here is one called BIP, and you can see the nice co-localization only for the deletion, not for the wild type. And through RT-PCR, we looked at a number of ER stress markers, and we saw that many of them were upregulated, and this is in pancreatic cancer cell lines. So if we look at the ER stress unfolded protein response, we saw upregulation of BIP, of PERK, of spliced XPP1, and CALR. So all of these are ER stress markers. At this point, we had a pretty good idea that probably what influenced the increased risk was ER stress and probably inflammation. And as I told you early, pancreatitis is a risk factor for pancreatic cancer. So this is what we thought was going on. So we then set up a collaboration with a colleague of mine in Madrid, Spain, where we created a mouse knockout model for this deletion. So mice only have one CTRB gene, CTRB1, whereas humans have both CTRB1 and 2. So we created this deletion of CTRB1 in the mouse, and we see reduced chymotrypsin activity. We see sort of histologic changes are not great, but when we start looking at the CTRB1 deleted protein, we see it accumulate, so similarly to what we had seen in the in vitro models. When mice are then treated with compounds that increase pancreatitis, this is a really well-known way of inducing pancreatitis in mice, and this compound is called sertraline. And you can see that the wild-type mice, after pancreatitis induction, they recover quickly, whereas the deletion mice take much longer to recover, and there are signs of increased inflammatory markers. So again, really nicely validating what we had seen in vitro and even taking it further in terms of linking it to inflammation. So in summary, we had this locus on 16q. We had 10 highly correlated variants. We saw splicing as an effect that then led us to this deletion that seems to destroy the activity of the CTRB2 protein and cause it to be retained intracellularly. I didn't tell you, but actually there are also GWAS signals for type 1 diabetes and type 2 diabetes, and the deletion increases risk for type 1, but not for type 2, which is kind of interesting because type 2 is a risk factor for pancreatitis. Type 1 is more related to inflammation. So maybe this signal is working through inflammation. So I told you about the reduced function and what the knockout model showed us in mice. What we're doing now is that we're crossing these mice with KRAS mutant mice to ask the question, if you have both KRAS and this deletion, do tumors occur earlier? So that work is ongoing. So this is one of the signals. I want to tell you about a second locus on chromosome 5. And this is a locus that's really close to the TERT gene and the CLPTN1L gene that I told you about. What's interesting here as well is that this is not a specific pancreatic cancer GWAS signal. It is seen in most cancers that have looked, whether it's prostate, breast, bladder, lung, etc. So this is a real multi-cancer locus. A while ago, we looked at 6 different cancers to pull them all together and try to map comprehensively signals at this locus at 5P. And we basically identified 5 signals. And very interestingly, each of the signal has cancers associated with it, where one allele can increase risk of some cancers, but it can be protective for other cancers. And this is really something that nobody understands at the moment. How can the same allele increase risk for one cancer, but be protective for the next one? And here are the 2 genes. So TERT encoding the catalytic unit of telomerase, and CLPTN1L that I told you is overexpressed in pancreatic and lung cancer. Since this study, we've mapped the signal and worked on the signal over CLP. So going back here, it's that signal here. Increases risk of pancreatic and testicular cancer, is protective for lung cancer. So we basically, just to cut this story short, identified a functional variant here that's located about 17 kilobases upstream of TERT that influences TERT expression. So this is a very typical GWAS finding where the underlying mechanism is noncoding. So because of the other signals at chromosome 5, I teamed up with a colleague in my branch where we're looking at 4 different cancers, so pancreatic, lung, melanoma, and bladder cancer. And we decided to do a screen over this whole locus so that we could just comprehensively look at all SNPs in both genes and ask the question, is it TERT, is CLPTN1L involved, or is it very clearly TERT for all of them? So we first combined the GWAS studies from these 4 cancers. So for the cases, it's about 110,000 cases. The controls are close to 900,000. So first we fine mapped, we found 11 signals. Then we went in with something called MPRA, which is really just the luciferase assay in a library mode so that you can test multiple variants. And you hook a sequence up in front of a reporter gene, and one version of each SNP gets each allele. So you have an oligo with an A, an oligo with a T allele, a lot of barcodes so that you can do your readout by sequencing. Then we also used the CRISPR-I proliferation screen. So we first made sure that inhibiting the expression of both genes reduced proliferation, and that test was positive. So we basically said if we use CRISPR-I and just go across the whole locus with both genes and some surrounding area, if we see a decrease in proliferation, it's telling us that either TERT or CLPTM1L is probably involved. So that's how we basically got regulatory variants and enhancer elements or promoter elements. And then we put all of this together to identify multi-cancer functional variants. And that is shown here. So here we have these 11 signals, and we've colored them in different colors. And underneath here, first you can see how we annotated all the fine map variants. Again, we're looking at 11 different signals at the same locus. And we can go to public databases and ask the question, do they overlap histone marks? Thereby they might be gene regulatory. So this tells us a little bit, but when we added on top of that our MPRA results and the CRISPR results, we identified what we call functional variants. So when our criteria is met for significance and effect size, but we labeled 8 different variants as multi-cancer functional variants, and we took them further for more validation. So here is some of the validation that we did. You can see SNPs that have the same color, come from the same signal. So the purple signal, the red signal, black and blue. And this is from the MPRA, so gene regulatory activity. You can see the difference between the alleles. CRISPR effects on proliferation. You can see that they all have effects on proliferation. But then we went in and specifically targeted each of these SNPs with individual CRISPR, and here I'm blowing that up. So here you can see that the SNPs that overlap the TERT gene, so TERT is right here, promoter here, it's transcribed right to left. You can see that they all have effects on TERT expression. So probably these signals here are working through TERT. There's some effect on the other gene, CLPTM1L, but not very strongly. But then when we come to variants over here, we have 2 that show some effect, but not in both cell lines. But then we see this variant here that in some cell lines, and this is a pancreatic cancer cell lines, you see more of TERT. But in others, and here's a lung cancer cell line, you see less of TERT. So we're thinking maybe this is telling us that in the context of the pancreas or the lung, some of these SNPs may do different things. And of course, this is just the beginning. We need to do more work to understand this better. But this is really more at a scale helping us understand these signals. What we're planning next is to take a similar approach and go after all of the 119 signals that I showed you from the new scan. Interestingly, at this locus, we also came across a region in the CLPTM1L gene, and this is intron 9 of that gene. So we saw very strong effects on proliferation in this region, and this region overlaps 2 VNTRs, so variable number tandem repeats. They both have a 29 base pair repeat that are actually different from VNTR 1 and 2, but they're repeated multiple times. So this variant here, VNTR1, is close to 2.5 to 3 kilobases, whereas this one here is shorter. So what we did here was to, again, go in with PacBio long-read sequencing, make up a reference panel, and impute back into our GWAS. And then we saw sort of however we do it, whether we look at length in association with pancreatic cancer, different groups and clusters, this is really part of the signal overlapping CLPTM1L. And then, so this is the results from the imputation. So we see some VNTR links associating either positively or negatively with risk of pancreatic cancer. Here in A, this is one of the alleles. So this is an almost 3 kilobase VNTR. And you can see the different units by color. So the units are not all exactly the same. They can have 1 base per difference or 2 base per difference. But what they have in common is that they have multiple motifs with binding sites for a protein called TEF1 that is a member of the hippo pathway, signaling pathway. So this is not just once or twice. This is like tens of times that this is repeated. And here is the motif for that binding site. So when we use CRISPR-I to target the VNTR, we actually see downregulation of both TERT and CLPTN1L. So we believe that this might be a functional variant impacting both genes. Also, we see that this VNTR is a really strong enhancer. So you can see here how strong it is compared to empty vector or to our control. And one of the links might be stronger compared to the others, which is interesting. When we apply hippo pathway inhibitors, we see a dramatic drop in enhancer activity. So this is definitely being regulated by the hippo pathways. Then doing proteomics, we pulled down oligos that were labeled with biotin and identified some of the TEF hippo pathway factors as binding. And we could validate that with pull down in Western blots. So we believe that the hippo pathway transcription factors are binding this VNTR and helping it in terms of making it a strong gene regulatory element. So in summary for 5P15, so we fine mapped GWAS data from 4 cancers. We identified 11 GWAS signals, 116 variants that we then took forward to MPRA and proliferation-based CRISPR screens. And we identified 8 multi-cancer functional variants. And we saw at least 3 signals, if not possibly 4, that we can now account for in terms of the biology underlying risk. We have some of these signals where there are more than 1 SNPs that seem to underlie risk. And then in some cases, especially for this signal over CLP, we see both SNPs and VNTR underlying risk. So this really just tells us that the biology underlying these is very complex. It's not always gene regulatory. It's not always affecting coding regions like what I showed you for CTRB2. So we need to be really open to mechanisms and really just go kind of where the science leads us. So just to really stress that this is a work of a huge team in terms of the gene mapping studies. These are PIs from all the different studies. So this is really a worldwide effort that requires people with information and background in genetics, clinicians, biologists, and it's really been a pleasure to work on this for the last 18, 19 years. And then, I guess I had this after, no, there we go. And then these are some of the people in my lab. So June has done most of the GWAS and TWAS analysis. And then Ashley, Caitlin, Naomi, and Aiden have done some of the functional work. So Ashley led the work on CTRB2. Aiden led the work on chromosome 5. And again, the wet lab work is also a team effort. Mouse work, I've not really done any of it myself. I've collaborated with people who have that set up. And that's how we can take these findings further. So I'm happy, I know it's 5, I'm happy to take any questions.
Thank you. Questions? Trainees, any trainees with questions? And Dr. Zhang is eager as always to get a question or 2 or 3 in.
Amazing talk. Thank you so much for being here.
Thank
I
you.
guess I have more of a career question. Did you begin with GWASs? How did you get into this line of work?
Right.
And yeah,
Right.
it's amazing to integrate so many techniques, which is
Thank
how
you.
you led.
That's a really good question. So I guess even back in high school and college, somehow genetics always kind of interested me a lot. When I did my PhD, it was sort of, I was doing work on transgenic mice, so in a way genetics, but not quite. Also for my postdoc. And then this opportunity just came to join Decode, and they were doing all gene mapping. So I guess some of it is just chance, and some of it is like, this is really what I was interested in. And then coming back to the US, I think my experience in Iceland at Decode Genetics helped me a lot, because I think without that background, I would not have gotten the job there. So some of it is like really you're going where you want to go, but then sometimes like you get an offer and you just take it and you go with it.
Okay, before we get to faculty questions, I do want to remind recruits and trainees that the only stupid question is a question that you didn't ask but wanted the answer to.
They're always all good. No question is a bad question.
Excellent. Oh, okay, great.
Hi, thank you so much for a wonderful talk. I have a quick question because you focus a lot on, of course, fine mapping your GWAS results, and you quickly also mentioned polygenic risk scores, and I was wondering for cancer phenotypes, whether those large odd ratios for your single variants also translate to your polygenic scores, and how you translate that perhaps to clinic, or is the future really more in fine mapping?
Right, that's a really good question. I feel now with the bigger GWAS that we have, that we're much better set up for polygenic risk scores. We did indeed combine the 22, 23 signals we had. And I think our polygenic risk score for the top 5% compared to the median was a PRS with an odds ratio of 2.2, so not really great. What we're seeing now is quite a bit higher. It's still ongoing. We're not exactly sure of the final numbers, but it looks much, much better. And what we're actually working on at the moment that I didn't mention is that we're using, instead of just summing up our significant GWAS signals, we're using more sort of appropriate methods to look at all the data and generate polygenic risk scores. We actually believe that because pancreatic cancer is not very common, that if we focus on the population, we might have false positives and negatives, and it might not be good to take that into even clinical trials at the moment. But where we think that it might be very useful is in the context of high-risk mutations. So say if somebody is in a family and carries, say, mutation, inherited mutation, say in CHEK2, the odds ratios for pancreatic cancer is about 2. But what about if you have that inherited mutation and then a very high polygenic risk score? Will that push you up so that you should perhaps get more frequent screening for pancreatic cancer? So that's something that we're focusing on at the moment. And I think our study, even though it's smaller than many other cancer GWAS studies, like breast and prostate and lung, especially breast and prostate, have much higher numbers. But because we have so many controls, our effective sample size in terms of cases is actually around 60 or 70,000, if I remember correctly. So our statistical power is actually pretty good. So that's kind of where we're going with the polygenic risk score.
Thank you.
Okay, so yeah, you start from 119 risk low site, and finally you successfully get a chromosome 5 or chromosome 16. So the gap is really big. How do you decide that you really can choose these two low sites and to the study? Its study is really nice.
Right.
So just tell us, because everybody facing this kind of things.
Right.
The really big one,
Yeah, too small.
I mean,
Right. Actually,
Yeah.
you're right. Actually, the 119 signals, that is data from our new scan. So when we started work on chromosome 5 and 16 and these other ones that we worked on, we didn't have those 119. So we had, initially in our first scan, we had 1 signal. In our second scan, we added 3 signals. So when we decided to work those up, we only had somewhere between 5 and 22 signals. So when you have that number, if you see a locus with TERT, it immediately you are drawn to that. Loci close to genes that are important for pancreatic development, that also kind of catches your eye. So sometimes it's basically what is nearby in terms of genes at a locus, but then also with GTAGs, we can immediately correlate genotype to gene expression for all of these variants. And if you come up with a very strong eQTL, it already points to a certain gene being a likely gene. And for CTRB2, we did that. We didn't see anything, but at the same time, we were looking at splicing QTLs. And the CTRB2 splicing QTL was the strongest splicing QTL we saw. So sometimes it's the data that kind of pulls you, sometimes it's genes that you're interested in. But going forward with the 119 signals, we really need to just use massive screens because going one after one is not feasible. And I think going more broadly will sort of make the time from discovery to knowing something at least much quicker. So that's basically where we're going. We're looking at, we're using also more kind of appropriate cell models instead of cell lines. We're setting up organoid models. We're doing iPSC cells. We're incorporating ER stress. So we're trying to be a little bit more fancy in terms of the cell models that we use.
Thank you, thank you.
Sure.
Hi, I'm also a geneticist.
Great.
And so most traits, medical traits, have a heritability component and also environmental. And so I work in psychiatric genetics, and so we've done the GWAS, fine mapping, the functional annotation, all these things, and we're only getting so far without including environmental impacts. And so
Right.
I know that cancer also has environmental impacts. So just wondering, following on Jolene's question about polygenic risk scores, adding in environmental effects and interactions and things, are we getting there for clinical
Right.
utility?
Right, that's a great question. So we are with PanScan 4, we have collected sort of e-data for like BMI, of course, alcohol use, type 2 diabetes. So we have not started, but our next project will be G by E for exactly these things. So smoking, diabetes, obesity, and heavy alcohol use.
And all of those also are genetically correlated.
Yes.
So it's the fun interaction
Exactly.
moment,
Yeah. We have tried in the past G by E, but I think we were underpowered at that point. We may have found one new signal based on that analysis, but that was it. But I agree, I think we need to go there both in terms of GWAS discovery, but also with the PRS. So not only looking at high-risk genes, but PRS, and then also these risk factors. And we're pulling in the same information for type 2 diabetes, smoking, BMI, alcohol use, and we're actually using the UK Biobank. We've not started too much with all of us because it's smaller, but there's a really good database in the UK that has a lot of information. So we're doing our polygenic risk score analysis in that database because, of course, you want to do it not in the same samples you have, but in an independent data set.
We also have a question from one of our alumni. They've been zooming in this entire time.
Great.
Dr. Helena Greenblatt is asking, are there differences in percentages in ductal CA versus non-ductal?
In terms of frequency of the disease?
In terms of survival.
In terms of survival, yes, actually quite big differences. So PDAC, or pancreatic ductal adenocarcinoma, is quite lethal. The 5-year survival rate, maybe 10, 15 years ago was about 6, 7%. I think now it's closer to 10, maybe just above or just under 10. But endocrine pancreatic cancer is much, sort of the survival is much, much higher. So definitely endocrine cancer is less common, but people survive better. Pancreatic ductal is very, very lethal. Did that answer the question, you think?
She's on the chat.
She's on the chat, so
Okay.
So
I'm
let's
gonna
let's
say
yes.
Yes, okay.
Thanks again for joining us. Thanks for
Oh,
And Justine, anything else we should say before we disperse and head to the restaurant?
thank you all for attending Annual Research Day and doing such a good job in your posters and presentations. Thank you to the faculty for your participation and mentorship in providing feedback to our trainees.